The water here is below freezing, -2 degrees Celsius, 28 Fahrenheit. Cold enough to kill anything with blood, and yet something moves through it. A fish, alive. Its blood should be ice by now. It isn't. For decades, scientists couldn't explain how. What they eventually found broke everything we knew about survival. This is the Antarctic ice fish. It lives where nothing with a spine should survive. Water colder than freezing, permanently. Here's the problem. Salt water freezes at -2 Celsius, 28 Fahrenheit. But fish blood? It freezes at -1. That's a full degree warmer, which means every second this fish is alive, its blood should be turning to ice crystals. The question isn't how it stays warm. The question is, how does it stop freezing from the inside out? Antarctica wasn't always frozen. 50 million years ago, these waters were temperate. Forests grew on the coast. Fish thrived. Then the continent drifted south. Temperatures collapsed. The Southern Ocean became the coldest water on Earth. Most fish fled north, or died. But some stayed, and somehow, against all logic, they survived. Scientists first discovered this paradox in the 1950s. They caught fish in water that should have killed them instantly. The specimens were alive, healthy, swimming in liquid death. The researchers assumed their thermometers were broken. They weren't. The water really was below freezing, and these fish really were alive. Nobody could explain it. The first theory was simple. Maybe Antarctic fish had evolved lower freezing points in their blood. More salt, different chemistry. Researchers tested it. They measured blood composition from dozens of species. The results made no sense. The blood chemistry was almost normal, nothing special. Freezing point, -1 Celsius, same as any other fish. So the fish should freeze at -1. The water is -2. The math didn't work. The fish shouldn't exist. Then came the second theory. Maybe the fish avoided ice crystals by staying away from them. Pure, undisturbed water. It's called supercooling. Liquids can stay liquid below freezing if no ice crystal touches them to trigger freezing. But there was a problem. Antarctic waters are filled with ice, floating crystals everywhere. Contact is constant. The supercooling theory collapsed. These fish touch ice all the time. They eat organisms frozen to ice. They live in it. For years, scientists were stuck. The fish were alive. The physics said impossible. Something was missing from the equation. In 1969, a researcher named Arthur de Vries made a discovery that changed everything. He found something in the blood. Not salt, not sugar, something else entirely. A protein no one had seen before, and it was doing something impossible. The protein didn't lower the freezing point. It didn't change the chemistry. It did something far stranger. It stopped ice crystals from growing, physically, mechanically, like nothing biology had ever produced before. De Vries called them antifreeze proteins, but that name would turn out to be misleading. This wasn't antifreeze at all. Here's what antifreeze normally does. Ethylene glycol in your car, salt on winter roads. They lower the freezing point. Add enough antifreeze to water, and it won't freeze until much colder temperatures. Simple chemistry, well understood. But these proteins don't do that. The freezing point stays almost the same, -1.5 Celsius at best. That's still warmer than the water. So how do the fish survive? The answer required looking at ice itself. Ice doesn't form all at once. It starts with a tiny seed crystal. Then that crystal grows, molecule by molecule. In normal conditions, once a crystal starts, it spreads rapidly. Water molecules attach to its surface. The crystal expands. These proteins do something unprecedented. They attach directly to the surface of ice crystals. And they stay there. Picture it. A growing ice crystal, molecules trying to attach. And suddenly these proteins are in the way. They don't destroy the ice, they don't melt it. They physically block it from getting bigger, a molecular barricade. Scientists call this thermal hysteresis. The gap between melting point and freezing point gets artificially widened. The ice crystal wants to grow, it can't. The proteins are sitting on every growth site. Expansion is impossible. This was the first clue, but it raised a bigger question. Where did these proteins come from? How did evolution make them? Researchers sequenced the genes. What they found was disturbing. The proteins looked like nothing else in biology. Most evolutionary adaptations are modifications. An existing protein gets tweaked. A gene gets duplicated and altered. But these antifreeze proteins? They appear to come from nowhere. No ancestral gene, no clear evolutionary path. For years, this was the mystery within the mystery. How do you evolve a protein from scratch? It shouldn't be possible. Then scientists looked closer at the DNA and they found something that rewrote the textbooks. The antifreeze gene sits next to a digestive enzyme gene, a gene for breaking down food. Completely unrelated function. But parts of the sequences matched. Not the working parts, the junk parts. The non-coding DNA that supposedly does nothing. This was the breakthrough. Evolution had taken garbage DNA and turned it into a life-saving protein from nothing. It's called de novo gene birth, the creation of a functional gene from non-coding sequences. Extremely rare, and it happened fast. Antarctica froze over about 15 million years ago. That's when these fish needed this protein. 15 million years sounds long, but in evolutionary terms, that's a blink. Complex proteins usually take far longer. Evolution was under pressure, freeze or adapt, and somehow it pulled a working protein out of genetic garbage. But there's another layer to this mystery. Not all Antarctic fish have the same antifreeze proteins. They evolved separately. The icefish family has one type. The nototheniod perches have another. The Antarctic eelpouts have a third. All different. Same problem, same solution concept. Completely different proteins. Evolution invented antifreeze at least 4 times. This is convergent evolution at the molecular level. Different species, different genetic paths, identical survival strategy. And here's where it gets stranger. Some of these proteins don't just stop ice from growing. They have another trick. They can reshape ice crystals. Normally, ice forms flat hexagonal plates, clean, orderly structures. With these proteins attached, the crystals grow differently, spiky, irregular. The shape is fundamentally altered. Scientists realize the proteins aren't just blocking growth, they're selecting which parts of the crystal can expand. By sitting on certain faces of the crystal, they force growth only in specific directions. Ice becomes controllable. For decades, scientists thought they understood these proteins. Block ice growth, save the fish. Simple. But recent research revealed something nobody expected. The proteins might also be a problem. A ticking biological clock. Here's what happens when ice is blocked. It doesn't disappear. Tiny crystals stay suspended in the blood, forever. The proteins keep them small, microscopic, harmless in the short term, but the crystals never fully melt. Even when temperatures rise above freezing in summer, the ice remains. The proteins are too good at their job. Researchers discovered that Antarctic fish accumulate ice crystals in their tissues over their entire lives. Spleen, liver, blood vessels, tiny frozen particles that never go away. The fish are partially frozen, always. This was the twist no one saw coming. The adaptation that saves them might also slowly damage them. A Faustian bargain. The proteins stop catastrophic freezing, but they also trap ice inside the body permanently. There's no escape. And now scientists are asking a new question. If Antarctica keeps warming, will these fish be able to lose their ice? Early evidence suggests no. The proteins work so well that even in warmer water, the crystals persist. These fish evolved to survive the cold. They may not survive the heat. Their greatest adaptation could become their doom. The very mechanism that saved them from extinction might now prevent them from adapting to a changing planet. Evolution solved the ice problem, but it created a new one. And this time, there may not be enough time to solve it. The detective story ends with an open case. Life found a way to beat physics, but physics might still win in the end. What these fish tell us goes beyond Antarctica. They show us that life doesn't follow rules, it rewrites them. Evolution took non-coding DNA, genetic junk, and built a precision nanotool, a molecule that defeats ice itself. This happened not once, but at least 4 separate times. Different species, different genes, same impossible solution. It suggests that when survival demands it, life can innovate in ways we never predicted. The toolkit is bigger than we knew. Scientists are now studying these proteins for human applications. Cryopreservation, organ storage, frozen food quality. A protein that evolved in fish blood might one day help us freeze human organs for transplant without damage. But the lesson here is bigger. Life at the edge of possibility doesn't just survive, it invents new categories of survival. These fish didn't adapt to cold, they invented a technology, a biological machine that manipulates matter at the molecular level. The boundary between life and ice, between living and freezing, isn't where we thought it was. And somewhere out there, in the coldest, strangest environments, evolution is probably solving problems we haven't imagined yet. The question isn't what life can survive. The question is, what can't it? A fish that should be frozen solid swims through water colder than death. It carries ice inside its body, always. Evolution didn't just find a solution, it invented a technology we're still trying to understand. What else is out there, surviving in ways we haven't discovered? The answer might be stranger than ice.